So I wanted to thank Dean for coming to the Cancer Center Grand Rounds and for coming to Children's to be the Ely professor, visiting professor. I think I've known Dean for maybe 20 years. I'm not sure, but it's been quite a while. Our research is pretty similar to one another's. Dean did his residency in Loma Linda, his fellowship at Texas Children's Hospital, and then he moved over for quite some time to MD Anderson, and then more recently, about 6 years ago, moved to Nationwide Children, where he's the DeMarco Family Endowed Chair in Cellular-Based Therapy at Nationwide Children's and at Ohio State University. Dean has really made some significant contributions to the NK cell field. Prior to Dean's discovery, which I think he'll probably talk about, the field largely would isolate NK cells and grow them with IL-2 originally and then with IL-15. And Dean discovered that using a cell line that expressed membrane-bound IL-21 and 4-1BB ligand really made these cells grow in some very interesting ways, but also has allowed us to expand NK cells and not only study them in the laboratory, but use them therapeutically. Dean's a problem solver. He solves problems. If you look at his CV, I think he's got 21 invention disclosures and an additional 15 patents granted. He is on the scientific advisory board of many companies because he solves these problems and because he's visionary in his thinking. He's the PI on a number of foundation and federal grants. And I think maybe despite all of those accolades, I think the one that may be the best that really suits Dean is that he brings groups together. He has worked with people in South America. He has worked with people in the field of neuroblastoma, Children's Oncology Group, pediatric transplant and cell therapy community, among others. And so Dean is going to share with us some of the work that he's been doing and on NK cells, and I'm really looking forward to the talk. Thanks a lot for coming, Dean. And by the way, I'll say that I think this is the third year in a row we've tried to get you here, but because of COVID, we've been unable to. So thanks for the persistence.
Thanks, Mike. Yes, Mike and Leah have graciously extended and re-extended the invitation now, so for the third year. And it feels really weird to stand up here and talk without a mask and actually talk in public. So this is my very first public, actually traveling somewhere since the whole COVID thing started. So I feel like now Denver's going to have a special place in my heart for ending the mental part of it. So I'll talk about NK cells. I'll give a little bit of background about sort of why we think they're important. I consider myself a little bit of an accidental NK cell biologist, despite Mike's nice and glowing introduction. I was a T cell person and didn't really care about NK cells until a few things happened that I'll talk about. But we've now really kind of found a nice niche and a lot of clinical trials moving forward that I'm hoping we can move these on. So I've also had some really good meetings already during the day, and I, at the last minute, added some more slides to the deck because of conversations I had earlier this morning. And so I probably way overpacked the deck, and so I may have to talk fast to get through them, and we'll see if I actually get through, in time. All right, so. And sure, mine was all right. So this is one of the few diagrams about NK cells that actually put NK cells at the center of the immunologic universe. And so I use it all the time because most of the time NK cells are the forgotten poor cousins that nobody wants to talk about. In the graduate immunology textbook that we used at UT, it was 750 pages long, and there were a whole 5 pages that were devoted to NK cells and their biology. So the reason they're put here at the center is because not only and this is really speaking about cancer recognition, not only do they recognize and kill cancer, which is mostly what we'll talk about today, But they also have a very important role in crosstalk with the rest of the adaptive immune system. And so in that killing of cancer cells, they release antigens that can then be processed by dendritic cells. They release cytokines that can activate and mature dendritic cells and recruit CD4 and CD8 T cells and NK T cells. They can induce class switching and maturation of B cells for better antibodies. And all of this is done through that initial recognition. So their primary job is really on a day-to-day basis viral recognition, and they're the first responders to viral infections. And by doing so, they set up the inflammatory microenvironment that's necessary to have a robust T cell and B cell response. And so we might think about them as having the same kind of role in tumor immunotherapy. And I think what we'll see kind of towards the end of the talk is that it's not really about what's better, a T cell or an NK cell, but what role do they have and how they may actually interact together, and finding the right niche for each of these cells in cancer immunology. So the problem with NK cells in cancer is that, first of all, especially speaking of leukemias, NK cells are produced at a relatively constant rate by the bone marrow. And so you have marrow infiltration with your leukemia, and now you can't make NK cells anymore. So you lose some of your NK cells. And tumors learn to suppress things that further inhibit NK cells. They make TGF-beta. The enzyme IDO converts tryptophan to kynurenine, and kynurenine is inhibitory through the AHR pathway. There are some checkpoints that the classic ones for T cells, probably with PD-1 and CTLA-4, probably don't play a role so much in NK cells, but TIM-3 and TIGIT, and there's a whole bunch of other checkpoints that do that. So tumors suppress NK cells. And then we get somebody who comes to us as a newly diagnosed patient. We give them chemotherapy and radiation, and we wipe out every last NK cell that's left. So our therapy for cancer actually destroys the one cell that we have that actually natively recognizes and kills cancer. And so we think about this now as somewhat of a therapeutic opportunity. What places, what contexts within our current anti-cancer regimens do we have where we can restore NK cell function and augment what is already good therapy and make it even better? It's not, some of it is about trading toxic chemotherapy for cell therapy, but some of it is also about recognizing what we're doing to our immune system and how can we restore that. So most of the data that we have for efficacy of NK cells comes from transplant settings. A lot of it from AML, but there's some other solid tumor and even autologous settings. So the first real striking evidence was in this paper by the Italian group now 20 years ago that showed that if you had a mismatch between the donor and the recipient in the cure HLA direction, if there was no mismatch in those, 75% of the AML patients relapsed, and if there was mismatch, they had 0 relapses. It was a striking, this has never been replicated to this degree and with this dramatic a finding since then, but it still holds true across many other studies that this cure mismatch, which actually enables NK cells to see cancer as foreign, really does have a role in the transplant setting. So the number of NK cells matters as well, not just who they come from, but how many. And so in this study, they took and looked at how fast do NK cells recover in a transplant setting, and they somewhat arbitrarily split those that had by day 30, so they called this the NK30. If you had less than 150 NK cells per microliter in your blood or more, and just splitting patients across these two, the more NK cells you had less GVH, you had less non-relapse mortality, you had much greater overall survival, and this was mostly true for AML and a little bit true for ALL. So it isn't even just about the anti-tumor effect, it's actually about control of viruses for your overall, your non-relapse mortality, and it's about controlling GVH. So NK cells have, as I mentioned, this mismatch, and you can categorize the degree of mismatch by how many of the receptor ligand pairs you have. And so this was from Kathy Sue, and if you only have the BW4 that's missing, which is one of those groups, or you have one group, you have group 1 HLA-C missing, that's another one of the group, or group 2 HLA-C. So you have these 3 groups that you can have possibly missing. If you had all ligands present, then nothing's missing. That was the worst outcome. If you only had one missing, then you had this intermediate survival, and you can't have all 3, but you can have 2 of the 3, and if you had 2 of the 3, that was the best overall survival. So again, this idea of optimizing NK cell function on the basis of how they see the tumor seems to predict overall outcomes, particularly when it comes to disease-free survival. And then lastly, there's a group of activating KIRs that are highly variable across the population. And in this study, they showed that if you had both the 2DS1 and 2DS2 KIRs, you had much less relapse than if you only had one or the other or neither. So across number, inherited inhibitory pathways, inherited activating pathways, and reconstitution, all of those things regarding an NK cell recovery after transplant seem to predict better disease survival. And one more. So this is again with the activating KIRs. So while this study looked at specific activating KIRs, you actually have 8 of them to choose from. And you can variably inherit 0, or I think actually 7, 0 all the way up to 7. And so the more you have, we call you a B donor. If you have none of them, you're an A donor. And so donors who have more of the B content, which is those activating KIRs, those actually predict survival. So lots of different ways where we've predicted survival on the basis of NK cell function. So can we do something about it? And now this paper was actually a Korean paper. It was now 8 years ago, was one of the first ones to show that you might be able to do something about these outcomes if you just give more NK cells to patients. And they took and apheresed the donor who was going to get a bone marrow transplant. And most of the mononuclear cells went to the transplant, but they've siphoned off some along the way to prepare an NK cell product, and they gave extra NK cells at day 14 and day 21 afterwards. Small study, phase 1, historical controls, but in their historical control, they had much less relapse and higher overall survival when all they did was just give back more NK cells. So I'm going to take a quick little detour through some T cell biology to say, why does this matter for us as pediatricians in particular? And although I think it's important for across all cancers, particularly in pediatrics. And that's in taking a quick look at what you really need to have robust adaptive immune responses. So if you're looking at a T cell or B cell response, in particular T cell responses to cancer, what do you need? You need to have the presence of an antigen. So you have to have something that the T cells can recognize. You have to have adequate processing that antigen so that it gets onto the surface and the T cell can see it. You have to have presentation of that antigen in the right MHC. So you might have the antigen, but it doesn't fit in your MHC, or the MHC isn't present, so you've lost MHC. And you have to have a permissive environment, which doesn't have all the things that suppress T cells, or doesn't have checkpoint inhibitors or whatever else that is inhibiting the tumor. And you have to have enough of an immune system on the T cell side that can interact with all those, right? So there's multiple barriers then to having a T cell response. And all of these are areas that we are currently actively investigating and have therapeutics to solve. With everything from TIL generation, so that you generate the T cells, to epigenetic modifiers that upregulate MHC or force antigen presentation, or checkpoint inhibitors that block and help a more permissive environment. Okay, in just looking at checkpoint inhibitors, this is one way of looking at whether or not there's an available response. Is there actually something that your immune system can respond to in that cancer? And checkpoint inhibitors have now a good 10 years or so that we've been looking at them across many different diseases. And this study was really a meta-analysis of about 36 studies across response rates to checkpoint inhibitors and the relative median of somatic mutations in that tumor type. And what they found is that there was a really nice correlation, that the more mutations you had, the better your response rate to checkpoint inhibitors. Again, that's the antigen part. If you have antigen that you can respond to, then stopping the brakes of recognizing that antigen will help you respond to the therapy. If you're down on this end where you have really low mutational burden, and this is in the area, I want you to sort of look at what the actual numbers here, between 1 and let's say 5 mutations per megabase, your average response rate to checkpoint inhibitors is around 10%. So when we look at pediatric cancers, there's adult cancers on the right-hand side, and their median mutations per megabase is between 1 and 10 across these cancers. These are the most active and these are the least. AML is actually pretty low. So we would not expect AML to be a great responder to checkpoint therapy. The pediatric cancers are all lower than the very lowest of adult cancers, and on magnitudes of order less, log orders less. So where that lower level in that last slide was one mutation per megabase, the typical pediatric cancer is down closer to 0.1, and many of them are down close to 0.01. We don't have these high mutational burdens in our pediatric cancers that allow for good recognition of an adaptive response. In addition, not only do we not have the antigens, but if you have an antigen and you're lucky enough to get it presented, then you only need to get rid of that HLA in order to escape recognition. And what we found out looking through about 25 different pediatric cancer cell lines is that almost half of them had some downregulation of HLA. So even if there is an immune response, we're now selecting for ability to escape that. Either those that had downregulated expression, or those, in this case highlighted in the ones that have these purple blocks, are loss of heterozygosity. In this location is loss of heterozygosity at the HLA class 1 locus. So about 1/3 of the cancers completely lost HLA of one locus, and about 1/3 of the cancers shut down all HLA expression in pediatrics. All right, so back to NK cells. So one of the hurdles of giving NK cells, as Mike mentioned, is that we really didn't have a good way to make them. All we could do is put someone on an apheresis machine, collect as many of the white blood cells as we could, get rid of the T cells because they'll cause GVH, and activate what's left with IL-2. That was NK cell therapy for 10 years, from mid-1990s, maybe 15 years, to 2010, and maybe till now. There's still many groups that are doing it this way. Couple of problems. You can see here that the median number of NK cells delivered across, I have about a dozen studies that are on here, is about 2 × 10⁷ per kilo. And those of you who worked in a lab, you know that 20 million cells is like that in the bottom of a centrifuge tube, right? So for every kilo of your body weight, and many of these patients having actually pretty good bulk tumor, you've got a pretty small number of NK cells. And the cost of doing this is somewhere between about $8,000 and $15,000, depending on your institution and the donor and what you're collecting, because you have to do all the pre-testing on the donor. In some cases, you have to put in a line so you can do the apheresis. With the apheresis procedure itself, you have all the columns and beads to do your CD3 depletion and GMP costs of IL-2 activation overnight. And you can only really do it once. You can't do multiple collections, for multiple infusions, unless maybe you've spread them out over several weeks and your donor is actually willing to come back and do this over and over again. So one donor, one infusion, relatively small numbers. So what the field really kind of needed was, how do we actually get more of these cells? How do we generate large numbers of highly active NK cells that we can deliver over multiple infusions and higher doses? I mentioned I was a T cell person, and I kind of stumbled on this because we were trying to get our CAR T cells to grow with a feeder cell. And instead, every once in a while, NK cells grew. And so it became my job to actually figure out how to intentionally get the NK cells to grow. And what we fell on, rather unsuspectedly, was that this one feeder cell that had IL-21 on the surface was really able to work quite well. And the process became relatively simple. Just take peripheral mononuclear cells, add the feeder cell once every 7 days to keep stimulating them, throw it out, get rid of the T cells, and now you've got large numbers of NK cells. And what that actually led to, when we compared it with IL-15, which most of the world was looking at, was IL-15 would work for a couple of weeks, but then the NK cells stopped growing. They just flattened out, became senescent, and wouldn't grow anymore. Where when they grew with IL-21, they grew and grew and grew and grew. And on average, we can now easily get, in a 2-week period, we can get 3,000 to 4,000-fold expansion. We've had some donors that give us as much as 10,000-fold in 2 weeks. And in 3 weeks, we can get 50 to 70,000-fold expansion. Huge numbers, and somewhat donor dependent, but very consistent from a manufacturing perspective. It does change them a little bit too, and we found differences between IL-15 and IL-21. Along with this proliferative senescence, compared to freshly isolated NK cells, the IL-15 expanded cells would lose a little bit of their interferon gamma production. They'd lose a little bit of their TNF production. And and IL-2 and IL-6 were about the same. When you grew them with IL-21, they suddenly actually increased their interferon gamma production, maintained all their TNF, and now they even start making their own IL-2. So one of the reasons we thought this was really important is the most toxic part of the therapy of all those other NK cell studies was delivering the IL-2. It wasn't the NK cells that caused toxicity. It was the IL-2 you had to give to keep the NK cells alive. And then finally figuring out a reason for this, or perhaps, was we looked at the telomere lengths and found out that when you grew them with IL-15, they actually lost telomere length. And when you grew them with IL-21, they gained some telomere length. And so telomere shortening in ex vivo expansion of primary cells is one of the main reasons that you lose ability to proliferate. So for whatever reason, one that I'll show you later, we were able to actually maintain telomeres in the NK cells. So great, we can make lots of numbers. Numbers don't matter if they don't kill, so we had to make sure they actually are functional. And we have not found a single cancer type that NK cells can kill. They can kill lymphomas, leukemias, neurocrust tumors, neuroblastoma, melanoma, carcinomas, sarcomas, brain tumors, gliomas, medullo, PNETs, almost anything that we've tried, NK cells have been able to kill that kind of tumor. They don't kill every tumor line, and we don't expect that they will kill every patient's tumor. There are some resistance mechanisms that we still need to understand. But when normal tissue becomes cancerous, one of the hallmarks of that is upregulation of stress proteins, either because of metabolic stress or genotoxic stress. And those stress proteins are what NK cells recognize. So it's a common feature across tumors that NK cells are recognizing it's an abnormal cell, just like if it was a virus infected cell and was abnormal. So they're recognizing features of the cell, not actual antigens. And those are common. So this expansion approach generated something that we eventually started calling these super bright NK cells because they had high levels of receptors, really across almost all of their receptors, that they gained over time. And so this is looking at Cytoff spade plots, which allow us to look at about 30 different surface proteins at a time, and freshly isolated at day 0. After 1 week of expansion, 2 or 3 weeks, and you can see there's a sudden upregulation into populations, red being high levels of expression, into populations that weren't there before, and that it's really across the board. It's both inhibitory and activating receptors. That may actually be a good thing, although we don't want the inhibitory receptors to inhibit. It is necessary for licensing and activation of the NK cells. Death receptor ligands, maturation markers, and all of these other activating receptors. So they're quite different. So how do they actually look when they work? This is a neuroblastoma cell line stained with a green dye that will leave the cell when the NK cells encounter it and release perforin and granzyme, perforin which pokes a hole in the cell, and then the green dye comes out. So I think what's kind of important to see here is that in some cases you can follow, it's a little bit of a jerky video, but you can follow the arrow of an NK cell going from one tumor to the other and repeatedly killing tumor cells. And in the space of 4 hours, the NK cells are able to kill 98% of all of the neuroblastoma that was in this plate. This is a brain tumor. You see one, it's actually AT, I'm going to pause it just for a minute. This is an ATRT, which we all know is a very difficult pediatric brain tumor to cure. A huge cell compared to the NK cell. So this is the cell flattened out on the bottom of the plate, and this is the little NK cell. And you can kind of see it as it moves, trying to decide, is this a good cell or a bad cell? Does it have these stress ligands that I should be worried about? And then once it makes that decision and degranulates, it's really a matter of minutes before the cell death event happens. So degranulation rapidly moves these granules up to the synaptic surface, liberates perforin and granzyme, pops a hole in the tumor cell, allows the granzyme to go in and initiate apoptosis. So really very quick killing. Looking across a variety of pediatric cell lines, so we looked at the whole PPTP panel that NIH established the preclinical testing program that used very highly characterized cell lines that their past histories were known. And it covers a range of brain tumors and neuroblastoma and Ewing's and rhabdo, and there's some leukemia cells, some lymphomas. And what I want you to see is this is sort of average killing from 4 different donors of NK cells. And then the bar here is sort of average killing. There are some cell lines that are very resistant to NK cells. And we need to understand better why that is. Are there intrinsic resistance to the perforin and granzyme? Do they not have any of those activating ligands that NK cells seem to see? And there are some that are extremely sensitive, even as sensitive or more sensitive than what we consider the classic NK cell target that has no HLA on the surface and has no way to inhibit the NK cells. But what is not really apparent until you sort of look at the names and you know what they are, is that there are brain tumors down here, there are brain tumors up here. There are sarcomas that are down at the bottom, there are sarcomas that are up at the top. There are leukemias and lymphomas at the bottom, there are leukemia and lymphomas at the top. So like I said, there's potential for NK cells to actually interact with almost any tumor type. Individual ones, we need to understand better about why they may or may not recognize it. All right, so actually skipped something I forgot. I took out a slide that was probably important. One of the key differences between IL-15 and IL-21 is that IL-21 signals through STAT3, IL-15 signals mostly through STAT5. Both of these are signal transducers and transcriptional proteins that once they're phosphorylated go and turn on a transcriptional program inside the cell. But they have slightly different binding sites. So STAT5 will turn on one set of genes and STAT3 will turn on another. So we've been looking really hard to see why this whole process looks different between IL-21 and IL-15 on the basis of which genes get turned on by either STAT3 or STAT5. So the first was about this problem of senescence. Why do the IL-15 expanded cells undergo senescence? And I said, well, when we looked at it, their telomeres were short. Well, it turns out that telomerase, which is an essential gene for maintaining telomere length, was the most differentially expressed gene when we compared IL-15 and IL-21. And that's because STAT3 turns it on. So STAT3 with IL-21 gets to turn on the one gene that's important to maintain telomere lengths, which is telomerase. So we confirmed this when we looked at some pediatric samples of patients who have something called Job syndrome. And Job syndrome is a dominant negative mutation in STAT3. They don't have STAT3, so they can't, well, they have barely functioning STAT3. They have about 20% of normal function. And so if you can see comparing normal NK cells with NK cells from Job syndrome or T cells from normal individuals, or I should say healthy individuals in Job syndrome, in the early NK cells that are just coming out of the bone marrow, there's a huge difference in telomere lengths. The Job syndrome patients can't maintain the telomere lengths of their NK cells. But that's not necessary for T cells. When we looked at all the subpopulations of CD4 and CD8 T cells, we really didn't see much of a difference at all. It's really most important in the NK cell population, and perhaps because they're the ones that are constantly being made by bone marrow and have a relatively short life. So T cells have other mechanisms for protecting their telomere lengths. There's also some apoptosis and senescence genes that are important, in particular BCL2L11, which is also known as BIM, which is a pro-apoptotic gene. When we looked at NK cells in IL-15 or IL-21, there's a microRNA 124.3p, that is known to regulate BIM, and it turns out it's much higher in IL-15 and actually helps turn on expression of BIM, so you have much more of this pro-apoptotic protein. So all the other apoptotic proteins were relatively the same, whether they were freshly isolated IL-15 or IL-21, but this one, BIM, suddenly drops down very low when you grow it with IL-21, so they're protected from apoptosis. So there's a bunch of genes that change, and we're really just scratching the surface now of understanding all these different pieces. Suffice it to say that the actual transcriptional program of these cells changes over time, and even what is activated by STAT3 is not the same between a naive T cell and an expanded cell. And the overall genetic chromosomal makeup, the epigenetic modeling that happens in that, is not the same as any other cell that we know about in peripheral blood. So here we're looking at ATAC-seq that looks at chromosomal arrangements and protection. How much is the chromosome open to transcription? These are the most immature NK cells that we usually find in the thymus. CD56 bright NK cells that are the new immigrants out of the bone marrow. CD56 dim and ILC is up here, and the expanded cells are in a completely different population. They just don't look like other NK cells in the body. And the differentially expressed genes that are there are those around DNA metabolic processes and repair. They're rapidly growing, so they should be repairing and maintaining their DNA. Mitochondrial ATP synthesis, they're highly functional, and so they've got to have a lot of ATP turned on. But lots of these other regulatory pathways in biosynthetic processes, why they have the circadian clock rhythm turned on, I have no idea what that matters to NK cells, but we may find out that sleeping actually helps your NK cells, right? Get a good night's sleep, your NK cells are better. Glycolysis and the P53 pathway are really important, as we'll see in a minute, because they enhance metabolic fitness of NK cells. I'm changing slides on my computer, but it suddenly stopped up there. Oh, there we go. Actually, I already talked about this. I forgot it was on 2 slides. But the STAT3 itself, we think we have a little bit of an understanding about why that gets turned on when you grow the cells with IL-21. And there's a switch in cytokine production that are between a couple of transcription factors in the Ikaros family. And you switch from one that activates IL-2 to one that actually suppresses IL-2. But I'll skip over that. So another problem we've had with NK cell therapies is that they're really a bugger to cryopreserve. If you take just mononuclear cells from peripheral blood, B cells, monocytes, T cells, NKT cells, NK cells, all of them in there, and freeze them in DMSO in the lab, and then thaw them out, the B cells are fine, the T cells are fine, the monocytes don't care, and the NK cells are 2/3 dead. For some reason, something we still haven't really figured out yet, they don't like to be cryopreserved. And one of the things that has been proposed is that it's part of this glycolytic reserve, that in order to survive that process, you put a big stress on the cells, and they have to be able to then quickly ramp up glycolytic metabolic pathways in order to recover. And this particular group showed that if you enabled that glycolysis, if you cryopreserved the cells, that did a bigger hit on glycolysis, so you couldn't recover. So there's other clinical trials where they expanded NK cells with IL-15 and cryopreserved them, and then none of the patients ever responded, and they couldn't find the cells afterwards. And they did some other experiments to look at how they survived, and the IL-15 expanded cells couldn't survive the freezing process. So there became this dogma in the field that you just can't freeze NK cells. Not only will fresh ones not survive, but even your therapeutic cells won't survive. You have to give them fresh. Turns out that IL-15 actually induces senescence by the same pathway that makes freezing a problem. And so the IL-15 expanded cells still undergo this problem, and maybe even more so because they're further stressed by having this glycolysis trimmed down, and IL-21 actually enhances glycolysis. So before we knew that we weren't supposed to freeze the cells, we had already published 10 papers and treated 100 mice, and it was working, and we just didn't know that we were lucky and happened to have activated glycolysis in these IL-21 expanded cells. So here's sort of baseline rates. These are freshly isolated NK cells. This is glycolytic reserve, and the top bar is what happens when you put the NK cells in with IL-21. They just are suddenly able to mobilize these pathways. And this pathway is important for licensing. There's not very many things that we understand about why an NK cell becomes licensed or not, but at least one of them happens to be that they get tuned to be better in their glycolytic pathways. All right. So can we now, we've done a lot of killing of mice, done a lot of NK cells and plates. Can we actually get this to the clinic? One of the key pieces was to take this feeder cell as a genetically engineered cell line and create it into a master and working cell bank that had all the testing that the FDA requires for those kinds of clinical trials. That's represented down here at the bottom. The second was to try to figure out how to make this a reproducible process where we were having donor collections and GMP manufacturing and delivering of the NK cells. And so what we ended up with is we were going to send the donor down to the blood bank, let them make a Buffy coat of that unit, send the red cells back to the patient so they could get it when they were doing their bone marrow harvest, something that MD Anderson did a lot. We don't do as much. And then take the Buffy coat to the GMP, get rid of the T cells. This, we found out by a lot of trial and error that it's a lot more efficient to get rid of T cells if you deplete them at the very beginning than if you try to deplete them later. It turns out Miltenyi had a bunch of that data. They just didn't let people know. And so when you activate a T cell, they suddenly downregulate a little bit of their CD3 and they can't deplete as well. So had to solve that problem. We figured out a way to either infuse them fresh or cryopreserve, because we still had a lot of arguments about whether or not we could cryopreserve these, and then infuse them to the patient. So as we move this forward, we've got some questions to answer. We have sort of a basic platform, but we have to understand, like, what donor is the best? How do we pick the donor for this patient? We don't want to give them their own NK cells back. They have crappy NK cells and they didn't work the first time, so let's try to give them healthy NK cells. Although, I take that back. There was some data that you might be able to restore NK cell function of somebody's. And so we did do a couple of clinical trials giving their own NK cells back. Do we need apheresis? Can we get by? Are we going to get enough cells by just doing a blood collection? Like I said, with the prior approach, if you apherese, CD3 deplete, stimulate with IL-2, that was pretty expensive. And if we could get rid of the apheresis, we could automatically cut $6,000 to $8,000 off of the cost of the product. So could we get away with just doing a simple blood donation? Could we get enough T cell depletion for it not to be a problem for GVH? The FDA was asking us to not infuse more than 10 to the 5th T cells per kilo, which, if we were going to try to achieve 10 to the 8th per kilo doses, means we couldn't be more than 1/10 of 1% T cells in our final product. Can we do that? Don't know. It might be hard. Can they survive the cryopreservation? I don't think it's still an open question now. It was at the time that we were trying to design these. And can we design trials in a way that allow us to actually test that? And which feeder cell is the best? We think we already figured that out with IL-21, but is the feeder cell itself safe? This is a tumor cell line that we made into a feeder cell, and if there's any residual feeder cells, that could be a risk to the patient. So we had to demonstrate to the FDA that we'd gotten rid of all the feeder cells, or somehow didn't have to worry about the feeder cells anymore. So all those were pieces that we had to really address before we could move this forward into the FDA. So I'm going to move back a little bit. Along the same time as we're working on generating these NK cells, there's still some other trials percolating along with the old way, right? Put them on an apheresis machine and generate the NK cells. And the first transplant study that we tried was similar to the one that I showed you from Korea, where we thought we've got a lot of data. We've got some experience with haplotransplant. We had some experience with standard transplant, a lot of historical data to compare with. Can we just add NK cells to that transplant? And so this was the overall schema of that study. BuFlu conditioning regimen. I talked to somebody about BuFlu today. We got answers back from the FDA that were silly. Anyway, yes, you can do BuFlu. Spread out some space between the chemotherapy and the stem cell infusion, enough to infuse some NK cells and let them work, see if they can clean up residual disease. Everybody was worried that this third-party NK cell might actually cause engraftment problems. So we wanted to give ATG in between the NK cells and the transplant just to make sure. That actually eventually got dropped. There's never been an engraftment problem giving NK cells. But that's what the ATG was in there for. And we weren't really sure how long they would last or how long they were needed. So we just gave this one single infusion. Dose escalation study. Relatively small numbers, but we were able to at least stratify them across doses received. And the more NK cells a patient received looked like the higher their percent survival. Simple study. We don't know if it's meaningful, but at least we got some data, got things going. So our next approach was to take that same platform. Now that we can actually make large numbers of cells, can we take the same platform and now do expanded NK cells? So same BuFlu, same NK cell infusion, IO2, same ATG and stem cell. But now we wanted to compare different donor sources. What if we do a cure mismatch donor versus a matched sibling donor versus a cord blood donor? This was at MD Anderson at the time. We had a brand new cord blood bank. We had lots of cords available. We wanted to know cord blood versus other cell types. Well, the cell types part didn't tell us anything. We didn't have a large enough number of all those to actually differentiate them. But in terms of numbers, the expanded NK cells still gave us one more level of response. Again, not hugely significant in a phase 1 study with lots of different doses, but at least we felt like we were on the right track, that we were improving outcomes by improving NK cell numbers. So we decided to pivot to a different setting, which was the haplo setting, and specifically haplo with PTCy. And that was for 2 reasons. One, because we were already getting the NK cells from a haplo donor. So why not put it in a transplant that's also with that, rather than having third party? And then we don't have to worry about rejection, maybe. And the second is that there was some data evolving around the PTCy itself, that PTCy may be really harmful to NK cells. And so this was from an Italian group that showed that T cells in blue, pre-transplant levels at transplant or after PTCy, take a little bit of a hit from the PTCy, but the NK cells take much more of a hit and don't actually recover. At all times, the NK cells are lower in number than the T cells. And in terms of overall function, they didn't really respond. They didn't actually come back as normal, functional, and mature cells for almost 6 months after transplant. So we thought, now we've got an ideal setting. Can we infuse NK cells in the middle of this context? So the other piece that was important from this paper was that they identified that the better your NK cell recovery at day 30, the lower your relapse risk. So again, we knew that this was a setting where recovery mattered, and the better your NK cells recovered, the less you'd have relapse. So now we have something that we can fix. We can try to get faster recovery of these people down here by giving NK cells back. So this was the platform: Flu-Mel transplant, reduced intensity, stem cell dose, 2 doses of PTCy afterwards, and then follow-up. And we had about 120 patients we had treated on this regimen, and we wanted to exactly lay over the top of that the NK cell piece. Just 2 weeks before, we would harvest blood from that donor, make the NK cells for them, infuse them on day -2, day +7, day +28. Again, early on, because we thought that this was important for MRD, and we'd already stressed out the cells to make them more sensitive. After the PTCy, because this is probably going to eliminate all the NK cells we just infused, so they need more. And then another 3 weeks later, because we think the NK cells last for 3 to 4 weeks. So, and we can make enough, so we'll just do more doses. So this was also a dose escalation study. In the first phase 1 report, we actually had a significant reduction in case match controls, single institution, of relapse from almost 40% to less than 10%. And overall survival from about 65% to almost 90%. And all we did different was adding back the NK cells. In that phase 1 study, we also saw a decrease in GVH and in BK virus and CMV virus, which we weren't expecting. But if NK cells are really first responders to viral diseases, then maybe we should have. We should have thought about the fact that NK cells are important for CMV. Maybe this actually helps protect. Needs to be done more robustly. And so we moved this on to a phase 2 study, and we had a very carefully 4-to-1 matched controls with the CIBMTR database. Every patient that was on our study, we found as close a match as possible, 4 close matches as possible. And in that confirmed that now in 25 patients, the relapse rate, relapse rate was 4% if you gave NK cells, and in the 100 or so patients from the CIBMTR, their relapse rate was an average of 38%. So big reduction in relapse. Overall survival significantly improved. There was some intermediate that wasn't as much better as we were hoping for. It turns out we had a couple of deaths early on from really silly things, like a patient who couldn't get to the hospital because we were having a hurricane in Houston and died of bleeding because they couldn't get a platelet transfusion. But those things happen, and so you just have to take them right in a clinical trial. Immune monitoring studies allowed us to see that we had actually significantly improved NK cell number and function compared to the historical controls. Surprisingly, I only want to point one thing out in the T cell side. At day 30, we saw a significant reduction in CD8 T cells in patients who got the NK cells. Like, why would the NK cells decrease your T cells? Well, we know that in that peri-transplant period, as you're recovering, that a lot of what drives T cell proliferation is the homeostatic cytokine IL-15. You've got lots of excess IL-15, so all the T cells that came along in your graft are now being pushed to proliferate. If you instead give a bunch of doses of NK cells, they soak up the IL-15. They're the ones that grow in response to that IL-15. And now you don't get non-specific expansion of your CD8 T cells, which might be why the GVH decreases. Because you don't need non-specific expansion of T cells. You need expansion of specific ones. You want your CMV and your adenovirus T cells to grow. You don't want the GVH causing ones to grow that are non-specific. So we might have some mechanistic information here. We've done some CyTOF to try to understand persistence. Now, unfortunately, in this context, the donor of the stem cells and the donor of the NK cells is the same. So we can't discriminate them by chimerism or any other HLA or other method. But we thought maybe we can differentiate them on the basis of their appearance, their phenotype. Remember, I showed you that when we do this expansion, they super upregulate all of their receptors and become these bright cells. And so when we look at the product, we can see all the NK cells are in this bright upper right corner. And if you look at healthy donors, that's not a cell type that's actually normally there. Most of the CD56 cells are down here. Most of the T cells are down in its bottom left corner. The product doesn't have any T cells, but when you look at the patient, they have both T cells down in this corner. They have some normal NK cells that are recovering, and they still have this super bright population that matches what the NK product is. It's about the best we can do of trying to prove to ourselves that these cells persist. But if so, then they're at least lasting 3 to 4 weeks after the infusion. And so we think that's long enough really to do the job that they're doing this way. And importantly, when we go back and you look at that study that was from the Italian group, where T cells dominated NK cells at every time point in the first month post-transplant, in these patients, NK cells dominated T cells at every point in the transplant. So we've now reversed reconstitution to being an NK cell dominant immune reconstitution after transplant versus the T cell. All right, that's probably most of the biology piece. I'm just going to run through a bunch of other clinical trials that we've tried and some of the outcomes, so I'll breeze through them quick. We've given multiple doses of NK cells after FLAG for relapse refractory AML. You give a bunch of chemotherapy. Great, you do something to the leukemia, but you also wipe out the NK cells. Can we restore the NK cells really quickly afterwards? The first patient treated on this, I think, is really kind of a good illustration. He had so many past treatments. He had initially been diagnosed and gotten 3 and 7, had an induction failure. Finally got in remission after 4 cycles of high-dose Ara-C, then relapsed. Mitoxantrone and etoposide, had a reinduction failure again to clofarabine and cytarabine, reinduction failure again to clofarabine alone. Finally got into remission with decitabine CAA, went to transplant. He was MRD positive, got a CNS relapse, got some cranial radiation, got back in remission, then had a bone relapse, got a DLI, and then got a whole bunch of other things and never gotten remission again. So wanted to go on this study. He came in with 93% blasts in his bone marrow. He got the 5 days of FLAG chemotherapy, 2 weeks of NK cell infusions. 1 month after the first NK cell infusion, we did his bone marrow, he was down to 5%. We said, great, you're at least in remission, go get a transplant. And his primary physicians at another institution said, well, we don't really think he's ready for another transplant. We're just going to watch him. Like, don't watch patients who have 5% blast, but okay. So anyway, a month later, he was down to 0.5%. And they said, well, he's doing really well. He's going back to the Y every day. He's swimming. He was in his 20s. He actually re-enrolled to go back to graduate school. 2 months later, he was down to 0.01%. He had an ongoing decreasing AML burden, right? That doesn't happen with chemotherapy. I don't know what caused this. Was it the NK cells themselves? Were they now sort of living and persisting long enough to continue removing his minimal residual disease? Or was there some crosstalk to adaptive immunity and now he actually had a T cell response to his disease? We may never know. We will never know. But at least we know that there's an immune component now that we were able to re-stimulate as part of just giving him NK cells with the chemo. As Mike mentioned, we've helped a group in Brazil bring this up. They've had a couple patients that they enrolled with CNS AML. We excluded those patients in our trial. This patient in particular had leptomeningeal disease around the brainstem and the lateral horns. This is one MRI 1 week before treatment, got his chemo and the 2 weeks of NK cells. A week after the last NK cell infusion, he came back into the hospital with slurred speech, ataxic gait, mental status changes, went to the ICU, got this x-ray where he had this broad inflammation around the same areas where he had residual disease, got some steroids, got better, in complete remission for about 9 months after that. So we think the NK cells can probably cross the blood-brain barrier and go to sites of inflammation, sites of active disease. In case they can't get to the brain, we've actually tried infusing NK cells directly into the brain. So we expanded NK cells from patients with brain tumors, generated a large bank of those cells. We can't infuse a lot, but we had the surgeon leave an Ommaya catheter in the resection site after recurrent resection. And can we infuse like little amounts 3 times a week? That ended up like driving everybody nuts. So when we started to dose escalate, we said, okay, we'll just do it once a week. But we were able to, in some patients, give 27 infusions into their brain of NK cells and had minor toxicities. One kid had some headaches. One kid had a little bit of ataxic gait for a day that went away by itself. Not great responses, but at least some stable disease that we were able to see. And this was with autologous NK cells, which we wanted to do because we were really worried about putting foreign cells into somebody's brain until we tried it first. But we really think that we might be able to do better than this if we can infuse larger numbers of cells and more healthy cells than just patient's own cells. All right, so barriers here. How do we move this forward now? This is pretty expensive, especially when we're making a new NK cell product for every single patient. It's not quite as expensive as CAR T cells, but we're still probably $25,000 to do an expansion. Some of the patients give poor expansions. Some of the donors give poor expansions. We can't predict. So, there's a lot of variability from person to person. The timing and resources are difficult. We have to time a GMP spot to right when a patient needs it. So lots of things that don't work very well. So we've really been working on how do we translate this now to a universal donor approach. We've done autologous NK cells. We've done haploidentical NK cells. Can we actually think about identifying the ideal NK cell donors, making large banks of NK cells from them, and having frozen, ready to infuse cells for any patient that comes in? So what is that optimal donor look like? From the original slides I showed at the very beginning, that would be someone who has cure ligand mismatch. So they have the best alloreactivity. Their cells are licensed. They have a high proportion of activating receptors. They have NKG2C expression, and we can do that. We can identify those donors. So we've worked for the last 2 years with the Be the Match Biotherapies, the biotherapy arm of the NMDP, to use their database to identify people that we think will meet these criteria, collect a vial of blood from them, make sure that we can actually expand their cells, that they have a good number of NK cells and expand well, and then do an apheresis collection, aliquot that. And now we have enough to actually generate enough for 80 to 100 infusions of NK cells from a single donation. And the cost goes from $25,000 a patient down to about 3 to 5. So huge cost savings as well as better ability to do it. So this is really just started. We started by trying to replicate the relapse refractory AML study. We've now treated 6 patients on this study, and we have some HLA. We can now target this by HLA differences, so we can get antibodies to donor versus recipient and follow them over time. No difference in their T cell component, but in the bottom right is the patient HLA type. In the top left is the donor HLA type, and you can see almost all, there's nothing of donor, like a few ditzels that come across. But then by about day 7, we've now shifted from being a half of recipient cells to half of donor cells. And this, they're just a prominent donor persistence that we can now find. I think really interesting, there's this population in the top right that is double positive for both donor and recipient HLA. And we think those are either doublets, an NK cell with a tumor cell, or NK cells pick off little bits of membrane from their targets, and it may have been trogocytosis of the donor HLA, but now you have like evidence there's actually interactions between the donor NK cells and the recipient. All right, 2 pieces I'll try to go through in 5 minutes each and still leave a couple questions for a couple of minutes for questions. what do we do next? How do we now we can get numbers. We think we've solved the manufacturing piece. We can cryopreserve them. We know what the donor type is like. We now have some tumor intrinsic microenvironment questions we need to overcome. We know, particularly for solid tumors, that TGF-beta is a really important cytokine. And even when you find NK cells in that tumor microenvironment, the TGF-beta has altered their function in multiple ways. They downregulate CD16, they downregulate NKG2D, it decreases production of granzyme. Their overall cytotoxicity is impaired. So can we make NK cells that are now TGF-beta resistant? We started this by saying, well, let's just throw a little bit of TGF-beta into the culture. That'll suppress the ones that are sensitive and let the resistant ones grow out. Not a bad hypothesis. It was completely wrong, but we'll get to that in a minute. So is it really important? And yes, so this is actually from over 1,000 cell lines in what they call the CCLE database at the Broad Institute, and their TGF-beta expression compared to normal tissue. And this is a log2 curve. So this is 1,000 times more TGF-beta than normal tissue. This is somewhere around 100 times more than normal tissue. And almost every single tumor type is represented in this. So TGF-beta is really common across all of them. Second thing you need to know when we look at the TGF-beta is how TGF-beta signals, which is through two halves of two different pathways. The canonical pathway, which is signaling through the SMAD protein. So these get phosphorylated and go and transcribe genes. And then there's a non-canonical pathway that we don't really understand very well, but has a whole bunch of other signaling pathways that activate. So when we threw in the TGF-beta to the culture, all the cells grew just fine. Turns out TGF-beta doesn't stop NK cells from growing. What it did do is it completely shut down SMAD3 expression. So we turned off the entire canonical pathway, and now the NK cells are no longer resistant, or no longer sensitive to TGF-beta suppression, and they've turned on a bunch of genes that they make even more cytokine than normal. And the SMAD3 suppression is at both the RNA and the protein level. So we've actually reconfigured the cells themselves to be TGF-beta. A whole bunch of genes that change. One thing I'll point out here is how hugely different they are from the regular NK cells that we expand. All of the gene variance differences account for 96% of the difference between these cell types. Donor to donor variation is very small, less than 4% of the variation. These cells actually happen to look a little bit like ILC1s. And one of the pieces of biology that's coming around, the innate lymphoid cells are kind of relatives of NK cells. And there's a whole group that migrate to the liver and in tissue that we call tissue resident NK cells or ILCs, depending on what you're studying. And we think that the TGF-beta in the local tissue might actually be doing some of the same reprogramming as what we observed. So what we accidentally did was recapitulate ILC development in our production rather than just making NK cells. They have huge granules, so we found out that they're actually a little bit better in serial killing. They can keep releasing perforin and granzyme over and over and over again. They have the same upregulated glycolysis, but they also now upregulate oxidative phosphorylation. So now they have two different metabolic pathways that they can use precursors from. And they've got changes in their addressants. Well, if they're tissue resident cells, that makes sense, right? Now they're actually cells that are supposed to reside in tissue, and now they've changed their chemokines and adhesion molecules to ones that are tissue resident. So again, kind of confirming this idea that they might actually be these tissue resident NK cell or ILCs, however you want to talk. So we want to get this moving in a couple of different trials. One of the first ones that was designed over at OSU was I got in touch with a breast cancer physician who was really kind of excited about having something for her relapsed patients. And after a lot of thinking and reading, we found out that the GD2 antibody that we use for neuroblastoma, dinutuximab, also known as DANYELZA, has been used in breast cancer as well. And it turns out the breast cancer stem cell is highly expressed as GD2, so it might be a really good target for this antibody. And gemcitabine happens to upregulate GD2, and the NK cells are necessary to mediate the ADCC of the antibody. So we had this crazy idea that if we use TGF-beta imprinted NK cells and the antibody in the gemcitabine, we can actually accommodate, one of the things the gemcitabine does is decrease MDSCs in the tumor microenvironment. So we can decrease suppression, activate the target, bind the target, and kill it all with this sort of circular synergy. So, so we brought it to the disease-specific study group, and they said, show us some proof. So we went back and did an animal model where we injected the animals with the MDA231 breast cancer cell line. We gave them the GD2 antibody alone. We gave them gemcitabine alone. We gave them NK cells and GD2, or we gave them all three together. And it was the one we gave all three together that was the clear outlier. Now, we didn't cure any mice of MDA231, but it's certainly a lot better than what we did with any of them alone. So I kind of ran through a bunch of the leukemia stuff, the most mature trial data, some of the things we want to move forward. Overall, just to say, we've now completed 140 patients on clinical trials and done a little over 400 infusions, and we have not yet seen a dose-limiting toxicity to infusing NK cells. They're very safe and easy to give. We're doing almost all of them outpatient. Those have been in transplant settings and non-transplant for leukemias across multiple institutions, multiple countries even. I mentioned the patients in Brazil, brain tumor patients, neuroblastoma. The current ongoing and planned trials are to now start to shift to our universal donor bank and see if we can get the same kinds of effect with less cost and easier delivery to the patient. And some of the other settings, like I mentioned with the breast cancer, a new trial in neuroblastoma, one in sarcoma, and in melanoma. So we have a lot of trials that are now coming along. For some fun, we found out a little bit by accident that we can grow dog NK cells. And as many of the pediatricians in here know, dog osteosarcoma is a really good model for pediatric sarcoma. So we've now initiated a dog study to do the same thing, grow a doggy bank of universal canine NK cells, infuse those during patient treatment, both around the surgical resection as well as the chemotherapy, and see if we can reduce the incidence of pulmonary metastasis. Just like kids, dogs die of their pulmonary mets, not of their primary disease. Let's get to the trial design. This is our first dog that we've treated. We've now treated 6 dogs, and we have in historical controls, 85% of the dogs have relapsed pulmonary disease by 1 year after diagnosis. And we now have 4 out of our 6 that have made it a year without pulmonary mets. Not statistically significant yet, but we're getting close. And then the last thing I'll mention is about, well, now that we finally have cells we can deliver, what can we do to genetically modify them? We struggled for about 8 years trying to get CARs into NK cells because we wanted to do the same thing with NK cells as we did with T cells. And NK cells just do not genetically modify the same way as T cells do. We found out a couple reasons for that. One of the reasons is that the lentiviral retrovectors that we use, the pseudotype, doesn't bind to NK cells. They don't have the same receptors on them. They look just like a T cell under the microscope. You would think they act like a T cell. There are critical receptors they don't have for the pseudotype, for the virus. And because NK cells are first responders to viral diseases, they have highly upregulated TLRs and other foreign DNA sensing mechanisms that they would rather die than be infected. So you put in foreign DNA and the cells die. NK cells don't like it. So we went instead kind of a backdoor approach. We decided to start by seeing if we could do CRISPR editing with Cas9 ribonuclear protein complexes. And in this case, you take preformed Cas9 as the protein, you incubate it with a guide RNA to make these complexes, and now there's just small RNA, which NK cells don't mind. They don't only mind if it's double-stranded RNA. And there's protein, which they don't mind protein either, and you can electroporate it into the cell. And so we did some proof of principle around this. Could we actually edit NK cells? And we were working with Gabriel Gower at Johns Hopkins, and he had the problem, he's an adult physician who treats multiple myeloma, that when you use daratumumab, you target CD38 on multiple myeloma, and you kill that by ADCC. So it's really dependent on functional NK cells. The problem is that NK cells also have CD38. And so instead, when you give patients daratumumab, they kill each other, NK cells, and you rapidly deplete all the NK cells that you need to intermediate the daratumumab response. So we wanted to see if we could get rid of CD38 and solve this. And just to briefly say, yes, it worked. We can get easily 80 to 90% efficiency of deleting the CD38. You can see these are the CD38 deleted NK cells. They kill better. These are the wild type and knockout without DARE. It doesn't change anything of their normal ability, but you actually restore their killing ability by getting rid of that part of the problem. And now that we can delete a gene, we can now think about knocking in one, because CRISPR allows you to now paste into your NIC a new gene into that place. And so you can do site-directed insertion. And so that actually was made possible by the fact that we had a GMP right across the street that was making lots of AAV vectors. And with that AAV vector, you put in homology arms on each side of the gene you want to put in, and the AAV can package that entire thing and deliver your gene as the knock-in piece. So we did a proof of principle with the mCherry. We're now working on generating CD33 CAR NK cells. And as you can see here, we have really high, nice 70 to 80% transduction efficiency, because the transient location of the AAV doesn't bother NK cells either. So you can electroporate in the CRISPR, then infect them with the AAV. The AAV eventually goes away, and now you have a nice gene insertion in its place. So we've done a variety of studies with CD33 targeting. We've got a couple of CARs to CD38, GD2 CAR, and we're trying to build these out now over the next year or two to enable some CAR NK cell studies. I'm out of time, so I'm going to skip this one. Just some fun CAR design stuff that we have that we're going to try to do. I will say 6 years ago, Nature Biotech said NK cells would be the next big thing in immuno-oncology. I don't know that I can claim they're the next big thing, but they're certainly exciting, and I think there's a lot of room for us to work on this in cancer therapy. And I have a lot of great people to thank, most of whom I hope I mentioned along the way. So thank you very much. And sorry I went over time.
I'm not sure if people have questions, if they can put them in the chat, or if there's people here who have questions. So I don't see any questions in this chat about CME. Anybody here, Jessica?
A little bit of a misnomer. So we used IL-21 for the dog, and we actually had to we had to check whether or not dog cells would respond to human IL-2, because I didn't want to have to remake the feeder cell. Yes. So they responded to the IL-21 fine, and we didn't remake that. We compared human and canine IL-2, and they did better with the canine IL-2. So we swapped out canine IL-2 for that. And in both of them, we grow them with the feeder cell, but we do need a small amount of IL-2 also. So we only use like 50 units per mil. It's a really low dose of IL-2, just enough to enable some of the other signals that they need. So the IL-21 signal by itself isn't enough. You need the IL-21 and a little bit of IL-2. Oh. Okay. And our measure was just how fast the NK cells grew. So human IL-2 might be better in some cases for what it's doing to the cell, and maybe it didn't work for growth because the human IL-2 actually worked too well or replicated higher doses, or something. But at least for expansion, the canine worked better. Yeah. Mm-hmm. I do, with one caveat. That's that if we take the same feeder cell that doesn't have membrane-bound IL-21 on it and we add in soluble IL-21, it doesn't work the same. And we think that that's because just like chronic IL-15 pushes cells to senescence, if you have constant IL-15 and IL-21 in the media, that's also not good. It does different things to the cell. What happened completely by accident when we put the IL-21 as a membrane-bound protein, the NK cell only sees it very transiently when it's attacking the cell. And then when it disengages, it no longer gets the IL-21 signal. And that pulsatile cytokine signal is probably more healthy for the cell. So we haven't tried it, and you're free to try it if you want. But I am not an IL-15 lover. I really do think that it does activate NK cells, but in terms of long-term persistence and function and avoiding senescence and exhaustion and all the other things that I showed you about metabolism and everything else, I think it has some detrimental effects that people aren't recognizing. The latest, so this is actually clearly shown in Jeff Miller's study, where they showed a difference between continuous infusion of recombinant IL-15 versus pulsatile sub-Q infusion. So they had to stop giving the IL-15, give the NK cells a rest, and then give it again. So they do it like once a week. If they gave it constantly, then it drove the NK cells to be dysfunctional. And then there's a recent paper, like just in the last month, by Chalice Bonifant at Hopkins, and she engineered NK cells to release soluble IL-15, and it killed the mice. So it provided better tumor control, but it also initiated toxicity to them, that the mice actually died sooner, even though they didn't have tumors. So IL-15 is potent. I think use it with caution.
I have a question. So the feeder cell line and membrane bound 21 or 1, and all the effects discussed largely. So how can you that 4-1 or what? How?
I'm completely convinced it's necessary. I don't know if it's the best. We haven't compared a bunch of alternatives. And I don't really know what part of its contribution there is. There's a whole bunch of separate data around 4-1BB signaling and what that does to NK cells that I've not made a primary part of my study. But there's been lots of variations of this feeder cell. We made it once as a bulk cell line. Then we made it as a clone. That's the one that went into the GMP and MD Anderson. We made it another time that we thought we'd do an HLA knockout that was with a collaboration with a company, and that one got canned. I made it a fourth time that it ended up getting sold to a company out from under me, and I couldn't use it. And then it's now the fifth version that we're using now. And other people have built ones along the way. So Fate has built their own. There's a Korean company that's built their own. I know somebody in China that's built their own. And a bunch of varieties. And people like exactly the same, or they built similar ones. And there was one report from a Korean group that they used a LCL cell line, lymphoblastoid cell line, EBV transduced B cell, and they only put membrane-bound IL-21 in it. And the gist of their paper was we didn't need 4-1BB. Well, that's because it's already on a B cell. So they didn't have to add the 4-1BB ligand to the feeder cell because they started with a feeder cell that already had it. But we did compare with and without, and without the 4-1BB, it did not work very well. We only know in mice. We haven't been able to get much. We have blood samples from humans, And we only know that they stay around somewhat in the blood. And part of our struggle is if you find them in the blood, is that good or bad? So we've made this link that persistence in the blood is associated with good outcomes, but I'd rather see them go where they're supposed to go, where the tumor is, right? Well, leukemia, fine, they're in the blood, but there are also other places where they need to go. We do need to have homing receptors for marrow, for lymph nodes, and the liver. And one of the key things that we've identified is the most consistent chemokine receptor they have is CXCL3, which should mediate migration to the liver. And in our mice, that's where they show up first. So we infuse them IV. A few get trapped in the lung. They go back out into circulation. And for the first 48 hours, all of our luminescence, whichever way we look at them, all of them are in the liver. And then they finally start to migrate out of the liver and go to bone marrow and lymph nodes and tumor sites. So I don't know if the same thing happens in humans or not. Could that mean that without making any additional changes, they would be really good for colon carcinoma? Because colon carcinoma, you don't die of your primary tumor, you die of your liver mets. I don't know. All right.
Okay, without any additional questions, I think I'll just thank Dean so much for coming. Thanks a lot.
Thanks.